Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology topics. This is Decomposition and Forensics, beginning with microorganisms and decomposition. In nature, decomposition occurs producing carbon dioxide and water, and this is mainly carried out by bacteria and fungi. The enzymes break down the components within the dead material, and this is part of the nutrient recycling process that are necessary for plant growth. For this decomposition to occur, there's some conditions that have to be fulfilled. The first one is we need warm temperature because the decomposition is going to be enzyme controlled. Also, water is required because these decomposition reactions are hydrolysis reactions, and the nutrients that are broken down need to be dissolved in order to be absorbed by the microorganisms. Also, oxygen could be available for organisms that carry out aerobic respiration. In topic 62, forensic science and the time of death. Forensic science is carried out to gather facts about criminal cases. These can involve identification of the victim as well as the suspect. They need to identify the weapon that was used to carry out the specific crime. They need to identify the time as well as the place of the crime. They also need to know the motive. Investigating the time of death combines eyewitness and circumstantial evidence, as well as using body temperature, rigor mortis, and forensic entomology. We begin with body temperature. Various factors affect the body temperature, and these include the mode of death, meaning how the person was killed, the fat level. This is because organisms that are fatter have greater insulation, and they're going to lose heat or temperature at a slower rate in comparison to organisms that are smaller. The surrounding temperature also affects the rate at which heat is lost from the organism that has been killed. The surface area to volume ratio is also important because organisms that have, or people who have larger surface area to volume ratio are going to lose heat faster in comparison to those that have a smaller one. Then clothing as well as coverings. Bodies that have been clothed lose heat at a slower rate. The next method used is rigor mortis, and this is about the muscle contraction or the degree or level of muscle contraction of the dead organism. This is going to vary depending on the time after death. Usually rigor mortis begins between 2 to 4 hours after death or longer. About 6 to 8 hours, it's going to take full effect. During rigor mortis, the muscles are in a state of permanent contraction, and that is because there is no ATP, which is required for muscle relaxation. There is evidence that drowning victims usually experience rigor mortis at a faster rate because they lose a lot of ATP when they are struggling to stay afloat. In topic 63, the process of decay. After death, gut movement is going to stop, and the catalytic enzyme within the gut are going to begin to digest the tissues. Also, the lysosomes within the cells are going to release enzymes, and these enzymes are going to carry out lysis. This is going to lead to the decomposition of the body before even other organisms do colonize the body. So the body is going to be changed, and this allows the decay process to go on faster. The next part is looking at the stages of succession, and this is focusing on organisms that colonize the dead body. The rate of succession is going to depend on the mode of death, meaning how the organism was killed or how the organism died. If it was a murder and there is exposed wounds, then the succession is going to occur faster because the eggs and other organisms that decompose the body will have greater exposure. The ambient temperature is going to affect the rate of enzyme controlled reactions as well as the speed of hatching of the eggs. So this is going to speed up the rate of decomposition if the ambient temperature is optimum for certain reactions to occur. If the ambient temperature is very cold, then the body is going to be in a state of preservation, so the decomposition is going to be slower. Organisms that carry out this decomposition include the gut bacteria, like we've already talked about. It includes flies like the blowflies, beetles, and carcass beetles. In identifying the stages of succession, usually forensic entomology is going to be carried out, and this studies insects that colonize dead bodies. So their eggs, their maggots, their pupa, these are going to be collected from the decomposing body, and this could be grown in the lab in order to identify which kind of insects they are, as well as to compare with their life cycles because, of course, most of these organisms, their life cycles are known. But if some eggs are already laid on the body, they can be collected, hatched into insects. Those insects can be identified, then we can use their life cycles in order to use them to estimate the time of death. During succession, colonizers, these are usually anaerobic gut bacteria, and these are quickly going to be joined by flies, for example, the blowflies. The flies usually enter open areas like the mouth, nose, eyes, as well as wounds, where they lay their eggs, and these eggs are going to quickly hatch into maggots. The maggots will feed on the tissue, breaking them down. These maggots are going to be pet, and they'll become adults, and the adults are going to mate, and the cycle is going to be repeated. As the decomposition continues, the tissues are going to be liquefied, and other organisms or other flies can colonize the body, like the beetles that are going to lay eggs on the carcass, and their maggots are going to feed on the larvae. Also, parasite wasps are going to lay their eggs in the fly and the beetle larvae. As the body becomes dry, there is less water, some of these colonizers are going to disappear and pave way for other organisms, and this is the process of succession. In the end, we're going to have organisms that are more adapted to break down connective tissue as well as muscle, decomposing until only bones are going to be left, and these are going to be dry bones. Next, we go to polymerase chain reaction. This is used to amplify DNA. It requires a DNA sample, the enzyme Taq polymerase. It requires primers. These are small DNA sequences that join the separated DNA before copying begins. And then there are nucleotides. These are going to be used in formation of the strand or the new strand of DNA, and the buffer that is going to be used to provide the correct pH for the process to be carried out. During this process, the mixture is going to be placed in a PCR machine, and it's going to be heated to 95 degrees Celsius. This is to break down the hydrogen bonds between the DNA strands. Then it's going to be cooled to about 50 to 60 degrees Celsius so that the primers can bind. This process could take about 20 seconds. And then it's going to be heated to 75 degrees Celsius so that polymerase or Taq polymerase can carry out the attachment of the nucleotides. This is going to be repeated for various cycles in order to gather or collect the required amount of DNA. Next, we go to DNA profiling. During DNA profiling, restriction endonucleases are going to be used to cut DNA at particular points within the introns, and these points are called restriction sites. So different restriction endonucleases have different restriction sites. After the process, we're going to have fragments of DNA that are of varying size, and then gel electrophoresis can be carried out on these fragments. The process of gel electrophoresis is similar to chromatography. The DNA fragments are going to be placed in wells in the agarose gel medium in the buffer. So we can see the wells are around here. This is going to be the agarose. So there is also going to be a buffer. As the DNA is placed here, remember DNA is negatively charged, so it's going to be towards the negative electrode. And as we place the power source, these are going to be charged positive and the negatively charged DNA is going to move from the negative terminal towards the positive terminal. So the fragments are going to be placed in the wells in agarose gel medium in a buffer solution. Some non-DNA fragments are also going to be added to the gel, but in a separate well, and this makes it easier to identify some unknown fragments. You could put the blood sample from the crime scene in one well. In the other wells, you can put separately blood samples from the suspects so that you can compare the position of the bands within the gel after the process. So this gel contains a dye which is going to be like ethidium bromide, and this binds to DNA fragments as they move through the gel, which is going to fluoresce in UV light for visualization of the DNA fragments to be carried out. Another dye is going to be added to the DNA sample. However, this dye does not bind to the DNA but moves through the gel. Its movement is slightly faster than the DNA. This allows for the current to be turned off before the DNA reaches the end. And because we have different sizes of DNA fragments, they're going to move through the gel at different rates. This is because their molecular masses are different. So the positioning is going to depend on the sizes. The concentration is going to determine the thickness of the bands. DNA profiles can be used in forensic science, usually in law and crime, to identify suspects. They can be used in family testing or familial testing to identify family relations, like paternity, as well as in inheritance claims. They can also be used in science in classification to identify closely related organisms so that they can be grouped. During this process, we'll look for similarities and differences in the coding and in the non-coding parts of DNA. So this brings us to the end of this video. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.